Hi students, today we are going to learn about meninges, cerebrospinal fluid and blood-brain barrier. First coming to the meninges of the brain. The brain is enclosed in the cranial cavity by three concentric connective tissue layers. They are pia mater, arachnoid mater and dura mater. These three constitute the meninges of the brain. See, this is the skin over the skull. This is the periosteum. This is the cranium. This is the dura mater. This is the arachnoid mater. This is the pia mater. See, we have seen that the brain and the spinal cord also enveloped within three layers of the membranes, collectively known as meninges. Okay. So, these are the coverings of the brain. From superficial to deep, there are three layers. They are dura mater, arachnoid mater and pia mater. Mater, in Latin, it is related to the meaning of mother. The dura, in Latin, it means hard. So, this dura mater is composed of dense connective tissue and adheres to the inner surface of the skull and vertebrae. The arachnoid is a thin wispy membrane that lies just deep to the dura and it is superficial to the pia mater. The very thin clear membrane. So, pia mater is also very thin and clear membrane. This pia mater will directly adhere to the surface of the brain and spinal cord. So, dura mater, arachnoid and pia mater. The chief function of the meninges is to protect the contents of the brain and spinal cord. Now coming to the spaces. That is, we have three meningeal layers. They will form three clinically significant spaces or potential spaces or cavities. They are epidural space, subdural space and subarachnoid spaces from superficial to the deep. See this is the bone. This is the bone of the skull. This is the epidural space. This is the dura mater. It is having tough connective tissue. And this is the subdural space. And this is the arachnoid mater. And this is the subarachnoid space. And this is the pia mater. Okay. So they will form three potential spaces. They are epidural space, subdural space and subarachnoid space. Now coming to the structure and function. The epidural space, which is present between the roof of the skull and the superior surface of the dura. It has arteries and veins to run. And the epidural space generally doesn't exist, but it will exist pathologically. So, if any pathological condition is there, it will be present. See here, this is epidural space. Here they are giving anesthesia, but if there are any pathological condition, fluid will be accumulated in the epidural space. Since the superficial layer of the dura, that is the periosteal layer, is securely attached to the periosteum of the skull. So, normally the dura has layers. Dura mater is also having some layers. The superficial layer is tightly attached to the periosteal layer. So, there is no space. But in pathological condition, any fluid can be accumulated in the epidural space. Normally, it has arteries and veins. This dura mater is composed of two layers. First one is the periosteal layer. It lies closest to the calvarium, that is skull bone. And second one is the meningeal layer, which lies closest to the brain tissue. The periosteal layer has fibroblasts and osteoblasts, and it has large quantity of collagen, okay, extracellular collagen existing within its intracellular space. So, it has fibroblasts, osteoblasts, in between that we have collagen. So, this will give dura its strength. These two layers are mostly fused. Okay, only when they are separated at venous sinuses and dural reflections. So only at the place of venous sinuses, that is veins, and dural reflections, these are separated. So two layers of the dura, that is outer one is the periosteal layer, inner one is the meningeal layer. Now coming to the dural reflections. Dural reflections refer to the places where two face-to-face meningeal layers descend into the cranial cavity and forms septa, and they will compartmentalize the brain. So two layers of the dura mater will descend down into the cranial cavity and they will form septa and they will divide the brain tissue. See this, the two main dural reflections are falx cerebri and tentorium cerebelli. This is a falx cerebri. Okay. This is a blue color one is a vein. This green color one is the falx cerebri. So this falx cerebri is sickle shaped and it is attached to the roof of the cranium and suspends down into the interhemispheric fissure, and it separates the right and left cerebral hemispheres. So between the two cerebral hemispheres we have falx cerebri. Now coming to the opening in the falx cerebri, that is tentorial incisura. So in falx cerebri we have an opening that is called tentorial incisura. Okay, sometimes it is also called as tentorial notch. So why it is present? It allows midbrain to pass into the middle cranial fossa. Okay, so to pass the midbrain into the middle cranial fossa, there is a opening in the falx cerebri. That opening is called tentorial incisura. Next, the U-shaped tentorium cerebelli. So this is falx cerebri. This is tentorium cerebelli. So we are coming to the next dural reflection. That is tentorium cerebelli. It is U-shaped. Okay, like this. This tentorium cerebelli. This is a U-shaped. And it runs transversely between the cerebellum and occipital lobes. So where the tentorium cerebelli is present, between the cerebellum and occipital lobes. Next coming to the falx cerebelli. It is a small midline fold of the dura mater projecting anteriorward from the skull and into the space between the cerebellar hemispheres. So between the two cerebellar hemispheres there is a dural reflection that is called falx cerebelli. So between the two cerebral hemispheres we have falx cerebri. Between the two cerebellar hemispheres we have falx cerebelli, and between the cerebellum and occipital lobe we have tentorium cerebelli. So these are the dural reflections. Now coming to the dural venous sinuses. So what are the paired and unpaired sinuses which are present in the dura mater? The unpaired sinuses are superior sagittal sinus, inferior sagittal sinus, straight sinus, occipital sinus, anterior and posterior intercavernous sinuses, and basilar venous plexus. The paired sinuses are cavernous sinus, superior and inferior petrosal sinuses, transverse sinus, sigmoid sinus, sphenoparietal sinus, and petrosquamous sinus. So, in some sinuses are shown here. So, this is the superior sagittal sinus. This is the inferior sagittal sinus. Okay. This is the superior petrosal sinus. This is transverse sinus. This is straight sinus. So some sinuses are shown here. So there are 7 paired and 7 unpaired sinuses in the dura mater. The dural venous sinuses form the primary drainage pathways of the brain. So why these sinuses are important? Because they will form the drainage pathways from the brain. Ultimately, these will join the internal jugular vein through the sigmoid veins. Okay. So they are situated between the two layers of the dura mater. So dural sinuses are lined by the endothelium and their walls are thick but devoid of muscular tissue, and they have no valves. So they have covering of the endothelium and the walls are thick but there is no muscular tissue, and there is no valves in the dural venous sinuses. So they will receive the blood from the brain through the cerebral veins and the cerebrospinal fluid from the arachnoid space through the arachnoid villi. So they will receive the what things? CSF, and they will also receive the blood from the brain. Next, coming to the subdural space. So we have seen the epidural space, dura mater, then we came to subdural space. Okay. So this is the subdural space. The subdural space is a space which exists between the meningeal layer of the dura mater and arachnoid mater. So similarly, this space won't exist normally, but if a pathological condition exists, this space will be filled with the blood or a fluid. So these are the two layers of the dura mater, periosteal layer and meningeal layer. So between the meningeal layer and arachnoid matter, we have what is this? Subdural space. This is the subdural space. This is the dura mater, this is the arachnoid matter. We have subdural space here. Next, coming to the arachnoid matter. The arachnoid matter lies between the dura and the pia mater, and it is avascular membrane, and it is involved in the cerebrospinal fluid metabolism via the subarachnoid space, and it bridges the contours of the cortical sulci, and it is thin and lucent membrane. So we have no blood vessels here. It is a thin and lucent membrane. Okay, it is present between the dura and the pia mater. So this is the dura, this is the pia. In between we have arachnoid matter. Its structure consists of superficial mesothelial layer below the dura mater and a central layer composed of the cells which are joined by many junctional proteins, and a deep layer of less tightly packed cells with many collagen fibers within their intracellular space. So dura mater is having two layers. Here arachnoid matter is having three layers. What are they? Superficial mesothelial layer. Central layer has many cells. They are tightly attached by junctional proteins. And a deep layer has less tightly packed cells, but we have many collagen fibers in between the cells. And the clusters of arachnoid villi. So, arachnoid villi, many arachnoid villi together called arachnoid granulations when we can see it with the naked eye. So, these are the portions of the arachnoid that protrudes into the dura mater, and it serves as a network for communication between the systemic venous system and the CSF. So, these are the arachnoid villi. So, many villi together constitute the arachnoid granulation. So, this is a arachnoid matter and it protrudes into the dura mater. So, with the help of this, the CSF is going to the venous system. That is that in the dural venous sinuses. Next, coming to the subarachnoid space. The subarachnoid space is the cerebrospinal fluid filled space that exists between the arachnoid and the pia mater. So the primary functions of the CSF is to cushion the brain and spinal cord from trauma and to supply nutrients and to remove the waste products. In addition to the CSF, the major arteries of the brain run through the subarachnoid space. Also projecting into this space are arachnoid trabeculae, which are strands of arachnoid matter connective tissue. So, in subarachnoid space, we have all these things, major arteries of the brain and CSF and arachnoid trabeculae. Next, coming to the pia mater. It is the deepest layer of the meninges. Okay, and it is also composed of two layers. It follows the contours of the gyri and sulci. The outer layer of the pia is the epipial layer. It has collagen fibers and mesothelial cells, okay, which will connect the arachnoid mater with the arachnoid trabeculae. The inner layer or the intima pia, it has elastic and reticular fibers and adheres to the outermost layer of the neural tissue known as the glial membrane. So, this pia matter is having two layers, that is epipial layer and intima pia. This intima pia is also called glial membrane. So, this is the arachnoid matter. If you remove this, you can see pia matter. The cerebral pia matter forms sheaths around the blood vessels. So, this is a blood vessel. So, this pia matter will form sheath around the blood vessels that enter and exit the brain perpendicular to the meninges, specifically which are coming from subarachnoid space to enter into the brain parenchyma. So, this sheathing creates some space, an interstitial fluid filled space. This space, that is perivascular, it is called a Virchow-Robin space. So, this Virchow-Robin space is present between the vessel wall and the pia matter. Okay. So, this is the vessel. This is the vessel pia. This is the cortical pia. This cortical and vessel pia layers will be joined. It is forming some space called Virchow-Robin space. So, we have seen the three meninges of the brain and potential spaces. Now we came to the embryology of the meninges. It is formed from perimedullary mesenchyme. So these meninges are formed from perimedullary mesenchyme. So in a newborn you can see all these things. This is dura mater, this is arachnoid mater, this is pia mater, this is the brain tissue. The dura mater is derived from mesoderm. So the derivations are different. The dura mater is also called as pachymeninx. It is derived from mesoderm. The arachnoid matter and the pia matter together called leptomeninges are derived from ectoderm, and these will form first. So the leptomeninges will be formed first than the pachymeninx. And the meninges primarily receive the innervation from the vagus nerve and trigeminal nerves, and also some cervical spinal nerves also contribute to the nerve supply. Coming to the arterial supply, the middle meningeal artery provides blood to the dura mater through its branches. Okay. And it has specific gravity between 1.005 to 1.008. And pH is slightly less than the plasma. That is, it has 7.33 pH, while plasma pH is 7.4. But it is isoosmolar with plasma. That is, it is having osmolarity of 289 milliosmoles per kg per water. And it is a watery solution similar in composition to the blood plasma. So, what is the composition of the CSF? It has 99.13% of water and 0.87% of solids. In solids, we have organic substances and inorganic substances. Organic substances like proteins, amino acids, sugar, cholesterol, urea, uric acid, creatinine, lactic acid is present. Inorganic substances like sodium, calcium, potassium, magnesium, chlorides, phosphate, bicarbonates and sulfates are present. So these are 8 and these are 8 in number. And lymphocytes are also present. They are 6 per 1 cubic millimeter of CSF. See this specific composition of the CSF. That is sodium in milli equivalents per liter. So all these ions are mentioned in milli equivalent per liter. That is sodium in plasma we have 145 milli equivalent per liter, while CSF we have 150 milli equivalent per liter, slightly more in the CSF. While the potassium, it is low in the CSF, that is having 2.9 milli equivalent per liter, while plasma is having 4.8. And calcium is also high in the plasma 5.2. Magnesium is high in the CSF 2.3. Chlorine is high in the CSF, 130. Bicarbonate is high in the plasma, 27.4. Lactate is high in the plasma, 7.9. Phosphate high in the plasma, 1.8. Proteins. So proteins and glucose are expressed as milligram per 100 ml. So protein are present 7000 mg per 100 ml in plasma, while 20 mg per 100 ml present in the CF, very much low. Glucose is present 95 milligram per 100 ml in plasma, while 60 milligram per 100 ml is present in the CSF. So with the help of this, we are going to know that the sodium and chloride are very much high in the CSF, while other things are more in the plasma, and the protein is much more low in the CSF. So in pathological conditions, CSF shows some findings. So with the help of lumbar puncture, we are going to remove the CSF from the individual. So if it is a SAH bleed, subarachnoid bleed, then the color will be xanthochromatic color if you take the CSF within 6 to 12 hours. And it is bloody, but it is having no clots. The pressure is increased, CSF pressure is increased. And here we will see the glucose. So glucose is a normal here, lactate is normal here, but proteins are increased. Proteins are increased, and we have to see for RBC in the case of bleed. So RBC are present, polymorphonucleocytes, that is neutrophils are present, and lymphocytes are also present. So in a bleed, you can see the contents, that is cells in the blood, and the protein is increased. If we see in multiple sclerosis, it is colorless, all are normal, but the protein content is increased. Minimally, it is increased. And we can see lymphocytes. Importantly, we can see the oligoclonal bands greater than equal to 2 in multiple sclerosis. In no other pathological condition, we are observing this oligoclonal bands. Next, coming to spinal obstruction. So if in there is obstruction to the spinal cord, then protein is increased. That is, protein will reach more than 500. Okay. So actually it is 20 milligram per 100 ml, but it is reaching. So depending on the bleed, the protein will be increased. And multiple sclerosis, the protein will be increased up to 50. In spinal obstruction, it will be up to 500. Okay, or more than 500. So you have to see the increase in the protein and how much it is increased also. And in spinal tumor, it will be altered color, xanthochromatic color, it is cloudy, and pressure is decreased. Okay, and protein is increased, and lymphocytes are also present. So pressure is decreased in a spinal tumor. Next, coming to the bacterial infection, viral, TB, fungal infections. So in bacterial infection, the color is white or yellow in color, and it is cloudy, the pressure is increased, glucose is also increased to half or less, okay, than serum glucose. So glucose is increased. How much is? And it is normally 60, but it is increased. And lactate is more than 35 milligram per deciliter. This is the lactate. Actually, it is 2.6 only, but it is reaching 35 milligram per deciliter. Okay. And protein is also increased up to 500. And there are many neutrophils, greater than 500 neutrophils, you can see. So in a bacterial infection, the CSF is cloudy, there is increased pressure, and under the microscope you can see neutrophils, and there is increase in the glucose level, lactate level, and protein level. In viral infection and TB meningitis, fungal meningitis, aseptic meningitis, abscess, infarction, in all these it is colorless. Okay, and the pressure may be increased. So in all infections, the pressure may be increased. And in viral infection, the glucose is normal, lactate is normal, proteins are slightly increased, and here you can see lymphocytes. So if you see neutrophils, that is a bacterial infection, and if it is a viral infection, that is increase in the lymphocytes will be present. And in TB meningitis, what happens? The glucose is decreased, lactate is increased, protein is increased, you can see lymphocytes. Okay, so the glucose is decreased in TB meningitis. TB meningitis. Now coming to the fungal meningitis, you can see lymphocytes, neutrophils, increased protein, increased lactate. Okay, and the pressure will be increased. In aseptic meningitis, you can see all normal, but we can see lymphocytes. In abscess, we can see neutrophils. And in infarction, you can see increase of protein and some amount of increase in neutrophils. Traumatic LP. In trauma, the trauma is causing, then what is the condition? It is bloody. Okay, and cells you can see same as the cells which present in the blood. Next, coming to the dynamics of the CSF, that is how much it is forming. That is total CSF volume in an infant is 50 ml and in adults it is up to 150 ml. The production rate is in infants it will be produced at the rate of 25 ml per day and in adults it will be produced at the rate of 500 ml per day, and it will exert the pressure of 9 to 12 centimeter of water in the infants, and in adults it will be between 18 to 20 centimeter of water. So it will exert the intracranial pressure of this much. You see this? This is the CSF volume 150 ml. This is the blood volume, that is also 150 ml in the brain. So these two are constituting 10% and 10%. And our brain parenchyma volume is 80%, that is 1400 ml. So this is important in Monro-Kelly doctrine. So if one thing is increased, the other things should be decreased, because our brain and these tissues are enclosed in a tight cranial vault. It cannot be expanded in the adult human being. Now coming to the formation of CSF. How CSF is formed? About 2/3 of the CSF originates from the choroid plexuses, which are present in the 4 ventricles, but mainly from the 2 lateral ventricles. The most 2/3 of the CSF will be formed. Then the additional small amounts of the CSF are secreted by all ependymal surfaces of the ventricles and the arachnoidal membranes also, and a small amount comes from the brain itself through the perivascular spaces. So these choroid plexuses, what are these plexuses? The ventricles contain capillaries which are derived from internal carotid and posterior cerebral arteries. So our ventricles is having capillaries which are derived from posterior cerebral and internal carotid arteries. The choroid plexus is a tuft of such capillaries projecting in the cavities of the ventricles, and it is covered by cuboidal epithelium of the ependyma. So like this, a capillary is present, for example, it is covered by cuboidal cells. So those are called ependymal cells. The endothelial cells of the capillaries are not flat as elsewhere, but they are granular and cubical and has more mitochondria and vacuoles. So what it does indicate? It will indicate the active metabolic processes occurring in these choroid plexus cells. So these are not flat, they are cubical and have more mitochondria and vacuoles. These choroid plexus project into the temporal horn of each ventricle, lateral ventricle, and the posterior portion of the third ventricle and roof of the fourth ventricle. So where they are present actually? In the 2 lateral ventricles, they will project into the temporal horns and posterior part of the third ventricle and the roof of the fourth ventricle. These choroid plexus are present. See this? This is the blood vessel. Okay. These are covering ependymal cells. Okay. See here. For example, this is a blood vessel or capillary. It is having lining that is endothelium. Okay. And it is surrounded by epithelial or ependymal cells. And what is the function of this physiology of choroidal plexus? That is, it produces CSF. Okay, produces CSF. which is containing glucose, oxygen, hormones, vitamins and ions. And here waste products and solutes are absorbed. So they will release these products and they will absorb the waste products. So removal of the waste products. Coming to the mechanism of formation of the CSF, there are two theories. First one is the filtration theory. It plays a minor role. We are not discussing this. Secretion theory, it is the main process. So how it is secreted? The secretion of the fluid by the choroidal plexuses depends mainly on the active transport of the sodium ions through the epithelial cells that line the outsides of the plexus, that is through the ependymal cells. So through the ependymal cells, there should be active transport of sodium ions. When the sodium ions are transported, they will in turn pull the large amounts of chloride ions, because sodium ions is having positive charge and chloride ions is having negative charge. So positive charge will attract the negative charge. These two together increase the osmotically active sodium chloride in the CSF. So osmotic pressure increases in the CSF and it will draw the water by osmosis. So less importantly, the small amounts of glucose and potassium bicarbonate is also moving into and out of the capillaries. So glucose will move into the CSF, potassium bicarbonate will move out of the CSF into the capillaries. Coming to the rate of formation, if the rate of formation is 0.35 ml per minute or 20 ml per hour or 500 ml per day in an adult individual. See this? This is ventricle. So CSF is present here. This is one ependymal cell. This is the capillary which is having blood. So what is the actual mechanism by which CSF is formed? We have carbonic anhydrase enzyme in the ependymal cell. So the activity of carbonic anhydrase. What it is doing? It is generating carbonic acid, that is H2CO3, by combining water and carbon dioxide. So H2O plus CO2 combined together, they will give H2CO3. Then they will dissociated into H plus ion and HCO3 minus ion. Okay. So that is a proton and bicarbonate ion will be formed. These will act as a driving force to the sodium hydrogen exchanger, and so this sodium hydrogen exchanger is present in the apical membrane. This is the apical membrane towards the ventricle. This is the basolateral membrane towards the capillary. So what happens? These two will give the driving force to the NHE, that is sodium hydrogen exchanger, which is present on the apical membrane, which exchanges sodium with H plus. Sodium is coming in and H plus is going out. And here anion exchanger 2. Here what happens? Anions will be exchanged. That is chlorine is taken in and this bicarbonate is going out. That is it is entering into the blood. Okay. From the from these ependymal cells. Next, what happens? Additionally, the basolateral sodium bicarbonate NBCN1, it is basolateral sodium bicarbonate transporter. It transports bicarbonate and sodium ions. Again, this bicarbonate will enter into the cell with the help of sodium ions. Okay. Then what happens? These sodium ions will be thrown out and potassium ions will be drawn in with the help of sodium potassium ATPase. And with the help of NKCC, that is sodium potassium chloride channel, it will throw out the chloride ions. And with the help of this also, water will be moving. The dashed lines, blue lines will represent the water movement. And water will also be moved through aquaporin 1. So from the basolateral side, from which transporter it is entering, it is not known. But it is going out into the CSF with the help of aquaporin 1 and NKCC1. And sodium it can also move like this. Okay. So because the sodium is moving with the help of sodium potassium ATPase, and so it is attracting the chloride ions also, and here sodium chloride ions will be present. and it will cause osmotic pressure and it will attract the water. The CSF formation is affected by the rate of CSF formation and the resistance to absorption through the arachnoid villi. So, under normal circumstances, we know that 500 ml of CSF will be formed per day. So, the rate of CSF formation will depend on the absorption through the arachnoid villi. So, this is the arachnoid villi. This should absorb the CSF. So, if it is not absorbing, then the rate of formation will also be decreased. Then it depends upon the cerebral blood flow. If cerebral blood flow increases, okay, what happens? CSF pressure rises because of the increase in the cerebral blood volume. So, if the cerebral blood flow increases, the CSF formation increases. And it also depends on the arterial blood pressure. This does not affect CSF pressure within the normal range because of autoregulation. However, it does produces a systolic diastolic fluctuation in the CSF pressure. Then coming to the venous pressure. So the pressure in the intracranial venous sinuses, how it should be? It should be lower than the CSF pressure. Okay, to allow the continuous reabsorption at the arachnoid villi. In pathological conditions, what happens? This venous pressure, if it is increased, then the rate of CSF formation will be decreased. So this will also line the choroid plexuses. Now what happens? The fluid secreted in the lateral ventricles passes first into the third ventricle. So these are the lateral ventricles. Now it is passing, that is the CSF is passing to the third ventricle through interventricular foramina. That is called foramina of Monro. Okay. Then some amount will be also be secreted from the third ventricle. Then total together will be flowing to the fourth ventricle with the help of cerebral aqueduct or aqueduct of Sylvius that is present in the midbrain. Then it passes into the subarachnoid space through three small openings. What are these openings? The foramen of Meninges, which is present in the median structure, which is present as the median structure, and two lateral ones. They are called foramen of Lashka. So foramen of Meninges is present in the median aperture. Foramen of Lashka, they are lateral apertures. The CSF is entering into the subarachnoid space through these apertures. Next, the subarachnoid space is deepest actually in the base of the brain. Its expansions constitute the various cisterns to be formed, the largest of which is the cerebellomedullary cistern or cisterna magna, which lies between the cerebellum and the medulla and extends downwards below the foramen magnum behind the spinal cord. So, the deepest part of the subarachnoid space is the cisterna magna. It lies behind the spinal cord. Almost all the CSF then flows upward from the cisterna magna through the subarachnoid space surrounding the cerebrum. From here what happens? The fluid flows into multiple arachnoidal villi that projects into the large sagittal venous sinuses and other venous sinuses of the cerebrum. So, from the ventricles, the CSF has come to the subarachnoid space. Then from the subarachnoid, from the arachnoidal villi, the CSF is filtered into the venous sinuses. So finally, the fluid empties into the venous blood through the villi. The movement of CSF is rather sluggish within the vertebral canal, and it is assisted by pulsations of the arteries in the subarachnoid space or alteration of the posture also. See, CSF is also drained back to vertebral venous plexuses and segmental veins. So, you can see here, if it is drained in the superior sagittal sinus, then it will go to the confluence of sinuses. These confluence of sinuses will receive the blood from inferior sagittal sinus through the straight sinus. Then confluence of sinus will open to the transverse sinus. This transverse sinus will also get blood from superior petrosal sinus, which is getting blood from cavernous sinus. And this cavernous sinus is also giving blood to inferior petrosal sinus. Okay. And this transverse sinus is giving blood to sigmoid sinus. The sigmoid sinus and inferior petrosal sinus will open into the internal jugular vein. So ultimately, it is drained. The blood and CSF will be drained into the internal jugular vein. Next, coming to the absorption of the CSF. So we know that arachnoid villi, that is arachnoid granulations, will take the CSF. So after bathing the surface of the spinal cord in the base of the brain, the CSF will pass upward over the convexity of the hemispheres to be absorbed into the intracranial venous sinuses. So we have seen that in the subarachnoid space is flowing and it will be absorbed through arachnoid villi. Small amounts are absorbed by perivascular spaces also and the spinal veins also. Arachnoid villi are small finger-like projections of the arachnoidal membrane. So this is one arachnoid villi. Okay, it is a small finger-like projection from the arachnoidal membrane through the wall of the venous sinuses. So conglomeration of these arachnoid villi. So many arachnoid villi, if they are joined together, they will form a macroscopic structure that is called arachnoidal granulation. See this? The endothelial cells covering the villi has a vesicular holes when we seen through the electron microscopy. Okay, and it has been proposed that these are the large holes enough to allow relatively free flow of CSF and dissolved protein molecules into the venous blood. So these holes are enough to pass the CSF through these cells. When the pressure of the CSF is high, the pores will open up and fluid escapes into the venous blood. But venous blood cannot enter into the villi. Why? Because first one is the pressure within these sinuses are normally very low. Okay. So the venous sinuses have low pressure. And if there is any rise of venous pressure, what happens? The pores will be automatically closed. So venous blood cannot enter into the CSF. Further, the colloidal osmotic tension of the venous plasma, venous blood is high, whereas the CSF is having very low colloidal osmotic tension. This also helps the transfer of CSF to the venous blood. So 3 points. Okay. Thus the CSF is absorbed via the arachnoid villi into the venous blood of the cranium and spinal cord. Next, coming to the functions of the CSF. What are the functions? It has these 5 important functions. What is the first function? Protection. CSF protects the brain from damage by buffering the brain. So the CSF acts as a cushion and protects the brain and spinal cord from damage. And second one is buoyancy. The actual mass of the human brain is about 1400 grams in weight. However, the net weight of the brain, which is suspended actually in the CSF, is equivalent to mass of 25 grams only. This allows the brain to maintain its density without being impaired by its own weight. So our brain should not be impaired by its own weight. So the CSF is acting as a buoyancy fluid. Next function. For chemical stability and homeostasis, the CSF maintains the distribution of necessary substances and a waste product between the CNS and the bloodstream. Okay, so it is helpful for a homeostasis and chemical stability. So it will give necessary substances and removes the waste of substances. And the fourth one is prevention of brain ischemia, made by decreasing the amount of CSF in the limited space inside the skull. This decreases total pressure transport of biomolecules to the brain. So actually the brain ischemia will be prevented because of the presence of CSF. Okay. So it will clear the carbon dioxide, lactate, and it will maintain the constant intracranial pressure. And clearing of waste. We know that it will remove the waste which is produced from the brain through the blood. For elimination, this blood will go to the kidneys. So 5 functions of the CSF: protection, buoyancy, chemical stability or homeostasis, prevention of the brain from ischemia, and clearing of the waste products. We are coming to the next topic, that is blood-brain barrier. What is this blood-brain barrier? See this? The principal components of the blood-brain barrier are, this is a capillary. So it is having a wall of the endothelial cell. So between the endothelial cell we have tight junctions. So these endothelial cells along with the pericytes and along with the astrocytic food processes, these three will constitute the blood-brain barrier. And other cellular components will also be present like neurons, microglia. Okay, so this is the brain blood vessel. So if you say, if you magnify it, you can see this is a blood vessel. This is having endothelial cells. There are tight junctions in the between the endothelial cells of the blood vessel, and this blood vessel is surrounded by a pericyte, and outermost is the astrocytic end foods. So these three mainly constitute the blood-brain barrier. So these tight junctions actually formed by transmembrane proteins. They are junctional adhesion molecules, one JAMS, junctional adhesion molecule, occludin, okay, claudin. So all these are present and they are linked to actin cytoskeleton. So thereby they will form most intimate cell to cell connection. These tight junctions are further strengthened and maintained by the interaction or communication of the astrocytes and pericytes within the brain endothelial cells. So how molecules are transported across the blood-brain barrier? So these are the transport pathways across the blood-brain barrier. So what is this 7 pathways? It may be passed through paracellular aqueous pathway. That is between the cell. Paracellular means between the cell. Between the cell you have actual tight junctions. But some water soluble agents will pass through it. Okay. So this is the blood actually. This is the brain tissue. So water soluble agents may pass through this like this. And second type of transport is transcellular lipophilic pathway. Transcellular means throughout the cell. So lipophilic substances, lipid soluble agents may pass through this like this. So but the transport is very less for all these things. Next, transport of proteins with the help of a carrier. Okay, facilitated transport. Glucose, amino acids, nucleosides will use this transport. And some pumps will be there, efflux pumps. These will throw out some material into the blood. Next type of transport is receptor mediated transcytosis. Like insulin and transferrin, they will use a receptor mediated transcytosis. This entire receptor, okay, will be transported along with the molecule and the molecule will be released here. Next, adsorptive transcytosis. So the molecule will be absorbed and it will be transported. Adsorptive transcytosis. Albumin and other plasma proteins will utilize this type of transport. Next, coming to the cell-mediated transcytosis. Monocytes and liposomes all will go through the cell-mediated transcytosis. So here what you can see, monocytes and liposomes together they are coming. So here they are separated, liposomes and monocytes. In the brain tissue you have pericytes, microglia, astrocytes, neurons. So these are the 7 pathways, transport pathways across the blood-brain barrier. What are the functions of the blood-brain barrier? It has dual function. That is, it has a barrier function and it has a carrier function. What are the barrier functions? It functions as a paracellular barrier. So this barrier is formed by tight endothelial junctions and restricts the free movement of the water soluble compounds. And also it acts as a transcellular barrier. That is made possible by low level of endocytosis and transcytosis. So there is a low level of endo and transcytosis and inhibits the transport of substances to the cytoplasm. And thirdly, it acts as an enzymatic barrier. So complex set of enzymes including acetylcholinesterase, alkaline phosphatase, GGT, that is gamma glutamyl transpeptidase, monoamine oxidases and other drug metabolizing enzymes capable of degrading different compounds. So it acts as a enzymatic barrier by having all these enzymes enzymes in the cell, and it also acts as a cerebral endothelium, and it expresses a large number of efflux transporters, that is ABC, and ATP binding cassette transporters. Okay, ABCs means ATP binding cassette transporters, etc. It is having. So it is a it is having function of efflux. It is functioning as a efflux pump. So these are the four barrier functions: paracellular barrier, transcellular barrier, enzymatic barrier, and the cerebral endothelium. And it has a carrier function also. What is that function? Nutrients to the brain and removal of the metabolites. So it provides nutrients to the brain, and it removes the waste products from the brain. And small lipid soluble molecules and blood gases like oxygen and carbon dioxide can diffuse passively through the blood brain barrier, where essential nutrients like glucose and amino acids require specific transport carriers or proteins in order to reach the brain. Now coming to the circumventricular organs. What are these circumventricular organs? These are the areas of the brain where this blood-brain barrier is weak, and it allows substances to cross into the brain in a freely manner. Those organs are called circumventricular organs. So these are the leaky regions. These leaky regions are isolated from the rest of the brain by specialized ependymal cells called tanycytes located along the ventricular surface close to the midline. So some circumventricular organs are pineal body because it secretes the melatonin hormone and neuroactive peptides, and these melatonin is associated with circadian rhythm, so it should be released into the blood, so it is not having a blood brain barrier. And second one is the posterior pituitary neurohypophysis, because it is also releasing neurohormones like oxytocin and vasopressin. These hormones will be released into the blood. And area postrema, the vomiting center, when a toxic substances enters the bloodstream, it will get to the area postrema and it triggers the vomiting. And subfornical organ, it is a chemoreceptive area and it monitors the blood angiotensin 2 level. It is important for regulation of the body fluids. So here also blood-brain barrier is absent. And VOLT, that is vascular organ of lamina terminalis. It is also a chemosensory area that detects the peptides and other molecules. And median eminence, it regulates the anterior pituitary through release of neurohormones. So in the pineal body, posterior pituitary, area postrema, subfornical organ, VOLT and median eminence. So these are the circumventricular organs where blood-brain barrier is weak. So based on that, we explain the blood-brain barrier. And so in some parts, the dye is present. So those are the circumventricular parts. Now coming to the another barrier, that is blood CSF barrier. So this is the non-brain tissue. Okay. Here this is the brain tissue. The blood-brain barrier, we have tight junctions like this we have seen. Okay. So this blood-brain barrier, where it is present in the production of CSF. So blood-brain barrier is present in all the brain regions. So anatomically, if you see blood-brain barrier, it is formed by endothelial cells of the cerebral capillary wall, and also basement membrane and perivascular foot of the astrocytes are forming blood-brain barrier. Whereas blood CSF barrier, where it is present between the blood and CSF, it is formed by endothelial cells of the choroidal capillary and basement membrane and choroidal epithelium. Tight junctions are present between the endothelial cells of the blood vessel. Here tight junctions are present here between the epithelia, that is ependymal cells. Here tight junctions are present. Okay. In the apical membrane. Location. Blood-brain barrier is present in the whole brain except the circumventricular organs. Whereas blood CSF barrier present in the choroidal plexus level, that is present in the two lateral ventricles, third and fourth ventricles. Surface area is 155 centimeter square of the blood-brain barrier, and 75, it is exactly half area is present for the blood CSF barrier. Here the blood flow rate is up to 1.8 ml per minute per gram, Here the blood flow is very much high, that is 4 to 6 ml per minute per gram. Efficiency as a barrier, if you see, the blood-brain barrier is highly efficient. Here the blood CSF barrier is also efficient, but functionally somewhat it is leaky. So you understand? So this is the blood-brain barrier present in the whole brain except the circumventricular organs. Here and in the blood CSF barrier, where the CSF is formed, that is in the ventricles at the choroid plexus level, we have blood CSF barrier. So how this barrier is formed, blood-brain barrier? With the help of tight junctions present between the endothelial cells of the cerebral capillary and the basement membrane, and perivascularly you have pericytes and food process of the astrocytes. Here, the CSF barrier is formed from tight junctions between the ependymal cells, that is at the choroid plexus level. Okay. So, in this class, we have discussed first about the meninges. So, there are three meninges: pyometer, arachnoid matter, and dura matter. From pyometer, arachnoid matter, and dura matter from below to upwards, and the potential spaces formed from them we have seen, and the layers of all these matters we have seen. Then we came to the embryology. The embryological part also we have seen for the meninges. And coming to the cerebrospinal fluid, second topic, that is what is the cerebrospinal fluid and what is the composition of it, and how the composition is differing from the blood, and in pathological processes how the composition is varied, that we saw, and how much is the volume and how much is the pressure exerted also we have noted down. Then we came to the formation of the CSF, how the CSF is formed from the ventricles with the help of choroidal choroidal epithelium. Okay, that is choroidal plexus, and there we have blood CSF barrier. Okay, blood CSF barrier. Then how it is circulated, so how it is entering from the lateral ventricles to the third ventricle, then to the fourth ventricle, then from the foramen of Magenta and foramen of Lashka, they are entering into subarachnoid space. Then they travel in the subarachnoid space and they will get the absorption from the arachnoidal villi and arachnoidal granulations. That is about the absorption of the CSF. And we have seen that when we seen the electron microscopic picture of the arachnoidal villi, we can see the pores. With the help of pores, these CSF will be leaking into the venous sinuses. But the venous blood cannot leak into the CSF, that is into the subarachnoid space. So we have seen the formation, circulation and absorption of the CSF. Then we have seen the functions of the CSF. So 5 functions we have seen. Then we came to the third topic, that is blood-brain barrier and blood CSF barrier. So what is this blood-brain barrier? So where it is present throughout the brain except the circumventricular organs. What are those circumventricular organs also we have seen. So why that is not present? Because these organs are releasing some hormones which are affecting the body. So that hormones should reach the blood. Okay. So there the blood-brain barrier is absent. Then we have seen the names of the circumventricular organs, and about blood CSF barrier also we have seen where it is present. It is present at the choroidal plexuses. It is formed from the tight junctions at the apical surface of the choroidal epithelial cell, that is ependymal cells. So with this we have completed meninges, cerebrospinal fluid, blood-brain barriers, that is blood-brain barrier and blood CSF barrier also. Next video is about cerebral blood flow.